Hello, everyone. Good morning, good evening, good afternoon, wherever you are. Welcome to Lallemand Brewing webinar Biotransformation 101, the fundamentals and practical tips for better IPAs. During this presentation, we'll have a strong focus on the practical side. So at the end of this webinar, you should have some great tips and tricks to take home with you to make better IPAs. My name is Eric Abbott. I'm a technical support manager and new product manager with Lallemand Brewing. And I'm here giving a brief introduction before passing it off to Scott Sharp-Heward, who is a technical manager with Lallemand Brewing in Oceania and based in New Zealand. Scott's background is in production and quality brewing roles in New Zealand and in the UK with over 10 years experience before joining Lallemand. He's qualified in winemaking with a Master of Science and also qualified through brewing through the IBD. He loves the technical side of brewing and working with customers to make fantastic beer. He has a very strong technical background coupled with what I would call a strong sensory flair that he uses with beer judging through the New Zealand Brewers Guild for many years. He has a strong passion for talking about New Zealand style beers as well. So I'm going to talk a bit about the IPA brewer's toolkit. There's many levers that the brewer has at their disposal for controlling the parameters of your IPA. Hop quantity, more hops gives you more IBUs. But it also gives you more precursors and more specific aroma compounds depending on the hop variety. Different types of hops have different levels of thiols, terpenes, and other flavor compounds that are important for your brewing. The point that you add them is very important. You get whether it's in the boil, early boil, late boil, dry hopping, or other modern hop addition techniques. The yeast strain is very important because it interacts with the hops in different ways depending on the specific yeast strain that you're using. And we've done a lot of characterization on our specific yeast strains and how they interact with hops. Nutrition levels and other types of additions such as enzymes or other inactivated yeast that can enhance body or reduce harsh or off flavors in the beer. And in general, integrating all of these factors throughout the brewing process to make the best IPAs possible. So in terms of optimizing biotransformation, optimal biotransformation comes at the intersection of hop selection, a specific hop variety, the hop addition point, and the yeast strain. The hop variety and the hop addition point will determine the amount of aroma compounds and aroma precursors in the wort, and the yeast strain will interact with these hop compounds to increase or modify the aroma profile. So I'm going to pass it off to Scott. Throughout this presentation, we're going to be we're covering the basics of biotransformation, some details about terpenes and thiols, mainly hop selection and how the addition point leads to different amount of terpenes and thiols as well as their precursors, how to maximize biotransformation by selecting a specific yeast strain, and with a strong focus on practical application and how to use this information to build better recipes and to brew better IPAs. So I'm going to pass it off to Scott right now. Throughout the presentation, feel free to ask questions using the Q&A button at the right of the screen. There's a little question mark you can click on there. We'll keep track of those questions throughout the presentation and we'll leave plenty of space at the end of the presentation to answer those questions. If you see a question that you like, feel free to upvote it. And yeah, with that, I will pass it off to Scott now.
All right, thanks, Eric. Thanks, everyone, and all my colleagues for tuning in my native time zone. That is much appreciated. Coming to you from 3 p.m. Friday afternoon, which is the future for some of you, I know. Well, for probably all of you. And just to Eric's point on what we're going to be touching on in this presentation, I think certainly in the past, it's been really easy for us all as brewers to treat biotransformation as sort of a bit of a black box. You put inputs into it and something comes out the other end, and sort of sometimes you're happy and sometimes you're not. And I guess a bunch of the material I'll be presenting was to try to help brewers with a bit of a framework for how to think about going about it to maximize what they want and avoid what they don't want. So to get started, I always like to start with the definition. For the purposes of this talk, biotransformation is the chemical modification made by an organism on a compound. Okay, and why are we interested in this? Why is it useful for brewers? Well, through it, we gain the ability to better utilize ingredients, to create potentially more unique and distinct products, and also can attain sort of more sensory intensity as well. So what is the importance of what we're going to be talking about in modern hoppy beer styles? And I guess for a lot of us these days, we're talking about modern hoppy beer. We're looking at juicier hazy pale ales, IPAs, double IPAs, and so on. And potentially also sort of modern West Coast beers. So we're talking about, as you can sort of see in this Brewers Association guideline, we're looking for medium high to very high hop character, basically hop aroma flavor. And the characters that we're specifically looking for are typically fruity. So as you can see, fruity, tropical, juicy, many others. And certainly if you scroll down to the additional notes, there's also, some secondary characters of citrus, pine, spice, and floral, which can be present, but are not primarily what we're looking for. Okay, so these are the compounds we're largely focusing on today. And so what compounds are actually key here? Well, that always helps to determine that before we then start talking about them. And, forgive me, this is an excerpt out of a paper, reference at the bottom there. But basically, the debate is what are the key compounds in determining sort of the key aroma compounds in dry hop beers. And these are somewhat agreed upon as some of the key compounds that you're actually looking for in dry hop beers. And so just to give you a bit of an idea, how I always like to explain some of these compounds to avoid things getting overly scientific is many of these compounds occur in hops or due to fermentation, but they can also occur in, for instance, fruits. They can occur in other plants. So this is why beer can smell like specific fruits. It's not one of those things that, when you're getting into beer and you go to a beer tasting and there's always sort of superlatives coming out. It's not that there's actually not some basis. It's just that you need to sort of get your eye in or your nose in, as it may be, into being able to sense them. And so based on a really interesting scientific paper that sort of was looking primarily at fruity attributes in beer, you can see that some of these compounds have characters such as sort of green, piney, resinous would be your sort of myrcene, humulines, caryophyllines, woody even. Now, terpene alcohols such as linalool, geraniol, nerol, citronellol, they're also key, but they provide other characters such as floral, citrus, particularly rose often is brought up. Hop esters, such as the esters in red. These can give characters such as stone fruit, light sort of pomme fruit, so apple, pear, sometimes pineapple, sometimes also rum-like a little bit as well. And finally, sulfur-containing thiol compounds such as 3SH or 4-MMP have also been identified as key hop aroma compounds in dry hop beers, and these giving characters such as sort of tropical fruit, so passion fruit, sometimes grape, sometimes sort of grapefruit as well, white wine sometimes as well. Okay, and we'll come back to that previous slide. So when you look at so a lot of these compounds are getting into your beer as a result of hop additions, and particularly the interaction between hops and yeast. And so this would give you an idea of a sort of average composition of a hop cone, which is what we would call dry weight, which is to say there's still some water in it, but it's largely dry, so it'd be kiln dry. And what we're interested in is the tiny little dark green sliver on the right, which is our essential oil fraction. Okay, and so this contains a lot of the potentially volatile and aromatic compounds that we want to sort of transfer to our beer to give flavor and aroma to our big fruity IPAs. And so as you can see, this roughly fits into 3 categories, which we'll talk about shortly. So the hydrocarbon fraction, sort of various chain length terpenes typically, and largely, which could include some of the more savory resinous characters such as myrcene, caryophyllene, pinene, which as you can imagine is quite piney. And these compounds tend to be sort of spicy, resinous, woody. They typically, they're likely to be found in dry hopped beer specifically because these compounds aren't being flashed out any earlier due to fermentation or due to hot side additions. The next fraction in the purple is the oxygenated fraction, which contains terpene alcohols, such as what we've already discussed, geraniol, citronellol. These tend to be fruity, floral. They're often quite key in overall hop aroma, particularly when we're looking for fruity characters. This fraction contains other compounds such as aldehydes, ketones, esters. We're not going to go too much into any of those today, though. The final fraction is our sulfur-containing fraction, which, as you can see, is pretty minuscule, the total amount of essential oils. And this is our potentially fruity thiols, such as 3SH, 3S4MP. These could be tropical, grape, sweat, overripe fruit as well, citrus. In this fraction as well, it's not all potentially good things. It's also potential off flavors such as sulfides, thioesters, which can give characters such as cooked corn, cheese, onion, garlic. And so for the purposes of our presentation today, we're largely going to be focusing on the terpene alcohol and the thiol fractions and what you can do about getting more or less of them. So first, I think it's worth sort of skirting over a few of the different types of what we could term biotransformation. Sort of historically, we typically were only really discussing this first one, which would be the freeing of bound terpene and thiol compounds. So they start off, they're present in some hops, and they can be in both free and bound forms. So they can be in a bound form that has no sensory activity. And so basically through the action of yeast, we can free up some of these compounds and, hopefully get greater sensory intensity in specific areas. And because I've mentioned both these things, it sort of gives you a rough idea of what's going on. Essentially, in terpene freeing, the yeast is producing an enzyme called glucosidase, which it actually excretes into the wort. That will then cut a bond, which will be between a bound terpene, as you can see, geraniol here, which is attached, in this case, to a sugar. And this is typically how you would find bound terpenes in hops, particularly. And the yeast, through this enzyme activity, will cleave that. We'll get a geraniol, which is suddenly sensory active, and the yeast gets a sugar. So it doesn't care about our hoppy beer so much. It cares mainly about the sugar molecule. And in thiol, it's for thiols, it's a very similar idea, except in this case, the thiol is not bound to a sugar. It's actually bound to an amino acid. And without going too deep into biochemistry, an amino acid is a component of proteins. So it's a smaller part of the overall metabolic structure of the cell. And so through beta lyase enzyme activity, which happens inside the cell, it can be split. And we can get a free thiol, and the yeast cell obviously gets an amino acid. We can also see transformation between compounds within the groups. And a great example of this would be the terpene alcohol geraniol being converted into citronellol, which has a lower sensory threshold and a slightly different sensory profile. Citronellol is nearly only present in beer through yeast activity. So it's one of those characters. And some yeasts appear to be better or worse at producing it as well. For instance, Pomona, which has recently been released by us in collaboration with the Scartman Labs, shows really, really promising sort of ability to do this. And the final one, which I'll just touch on briefly, is esterification. So some of these compounds, these thiols and terpenes, etc., can be esterified into an ester form. And why is it interesting? It tends to be interesting because the esterified form tends to have a similar sensory profile, but at a far lower threshold. So, for example, geraniol can be esterified into geranyl acetate, and geranyl acetate has 8 times lower threshold for sensory. So that's one of the reasons we'd be interested in that. That being said, we are largely going to be focusing on terpenes and thiols. There'll be some touching on the other two, but that's we're mainly looking at free and bound in this presentation. So to start off with, we've got these are sort of a selection of pretty common terpenes in beer. I've already sort of touched on some of the characters that these can give. Certainly geraniol, which you see this geraniol / nerol, they're functionally very similar. They're just an optical isomer. So it's the same chemical composition, but it's just arranged in a slightly different way. They have slightly different thresholds in beer as well. So particularly geraniol and linalool can certainly be free from bound. They can also be sort of converted from other terpenes, which might exist as well. Citronellol, as I've touched on, tends to be directly as a result of yeast activity. And terpenol is very similar to citronellol, but with a different starting compound. It's a little bit less interesting as it has a higher sensory threshold, and it's kind of more sort of straight floral. And I think I've got numbers in here, and we're talking sensory thresholds in sort of the micrograms per liter range. And I guess it's always interesting to have a comparison. So the fault compound acetaldehydes, that sensory threshold is 10,000 to 20,000 micrograms per liter. And we're talking for citronellol down as low as sort of 9, just to give you an idea of the sensory activity of these compounds. And so terpenes are interesting as well because they can often be synergistic with other fruity compounds. For example, lactones and thiols, which can boost sort of specific fractions of the sensory profile or the overall fruitiness. And geraniol actually has been found to also affect bitterness by smoothing its impression and sort of boosting the perceived sweetness you can get in beer as well. So terpenes can obviously be added already in the free form as well, which is sensory active already. These free terpene alcohols can then obviously be modified by yeast activity further, such as being esterified or being converted into things like citronellol. And we obviously also have bound terpenes being contributed by hops as well. And so, again, terpene precursors from hops are typically glycosylated, which just means attached to a sugar. And so just to go over it again, you've got this initial non-aromatic compound through yeast-excreted glucosidase enzyme. It cleaves this bond, frees up a simple sugar, and then also a nice aromatic terpene alcohol for us brewers. And so it's just kind of a schematic to get your head around it. So in this instance, the yeast is actually excreting the glucosidase, which is freeing up the bound precursor. And we have internal enzymes within the cell taking in the geraniol and converting it into citronellol. Why is this important? Well, it's probably good to know for, I guess, how to think about addition points. And so the next slide I'm going to show you is, I guess, probably the first really practical information. Basically, we know certain strains within our catalog that show greater glucosidase activity, basically through a fairly extensive literature search where I just pulled apart as much hop data as I could, all in peer-reviewed journal articles. I could basically identify which hop varieties are high in bound terpenes and high in free terpenes. So this sort of idea of this flowchart is a really easy, quick guide to try to direct brewers towards choosing ingredients to maximize some of these compounds, should that be what they want. So if you want sort of a really terpene-forward beer, you could consider selecting some of these high bound terpene hops and using them in either the whirlpool or as a very early dry hop. You can combine them with the yeast strains, which show the best glucosidase activity. This is by enzyme assay data, which we've done internally within Lallemand. And you can sort of see that some of the strains we've identified are BRY97, which is our American ale strain, New England, and Voskvik. So really quite different genetic backgrounds, but similar sort of ability to free some of these bound terpenes. Now, obviously, too, we have an exogenous enzyme, aromazyme, which, when combined with bound terpenes, has the potential to free some of them as well. So that could obviously be a great ingredient for if you want more terpene expression, but you don't necessarily want to select one of these yeast strains. And so then the idea is that the hops on the far right, as you can see, are high in the free terpene. So if you do everything you can with the bound terpenes and get as much release as you can during fermentation, and then you still feel like you want more, or you want to select a dry hop specifically for those characters, here's some examples of hops that would contribute that as well. And I should say as well, this isn't an exhaustive list by any means. I was trying to be fairly diplomatic when I was building this resource in terms of like selecting hops from different countries, different backgrounds, because I think that's much more interesting than focusing too much on any one country's produce. And as you can see down the bottom, citronella is a little bit different because it's created through biotransformation of free geraniol. We actually want high free geraniol early in the ferment. And I should say the sort of optimal, which is why I've sort of suggested either whirlpool or early dry hop use for bound compounds, is kind of we want those compounds to be in there and interacting with the yeast enzyme at sort of peak of fermentation. The highest point of yeast is probably when we're going to get the most enzyme activity as well. And so for citronella, the way we could approach that is actually by using high free geraniol hops in maybe a cool whirlpool or sort of very early dry hop. Then you could use something like Pomona to maximize the conversion into citronella. And just as an aside, I found an interesting study on the technique called dip hopping, which has sort of been around for a while. I first learned of it from a brewery in Portland, Oregon. And this is sort of a process of steeping hop pellets in hot water for a period and basically adding this slurry to the fermenter at the same time as the yeast. So basically during or immediately post knockout. And initially, I think the technique was intended to be able to utilize hops that you wouldn't typically use for their aromatic properties, as it can drive off some sort of flavor descriptors like medicinal or onion garlic or reductive. And anyway, there was a study published on this last year, and it showed some really interesting results. They basically made a small batch beer, and they compared the dip hop method with essentially just late hopping. So very comparable hop usage rates, which were around 4.5 grams per liter of the total batch size, or for Americans, I converted it here for you, 1.13 pounds per barrel. And that was Idaho 7. And sort of a couple of the interesting points to come out of it, you can kind of see the graphs, which I've pulled from that study. And they appear to highlight pretty marked increase in terpene alcohols, free terpene alcohols like linalool and geraniol especially. And the sensory component of the study noted that the olfactory and gustatory pleasantness of the beers were increased as compared to the control. So just another idea for brewers to play around with again. Now we are going to head into the section on polyfunctional thiols. We're just going to be calling them thiols for short, because my mouth will get even drier if I say polyfunctional any more times. Now, these are obviously of big interest. They are obviously only a fraction of the less than 1% of sulfur compounds which exist in hop oils. They exist in two main forms that we're interested in. Obviously, again, sensory inactive, so bound/precursors, whatever you want to call them, and also as sensory active free thiols. And so thiols are a bit different than terpenes in some ways. The vast majority of thiol precursors from hops are not converted during fermentation. So they're extremely poorly converted from bound into free form. And it's quite possible that these low conversion rates are actually not a bad thing. at higher concentrations, some of these otherwise pleasant compounds can start sort of taking on more undesirable characters like rotten fruit or sort of tropical fruit that can be polarizing, like durian or jackfruit. And just a note here from my colleague Carlos, who also shares in the Biotransformation sort of resource group. He spent some time as a winemaker also, and he noted that when winemaking in New Zealand, sort of thiols are present in a lot of New Zealand white wines. And often a way that wineries would approach it is they would make one tank, which was, for instance, one tank of a very, very high, basically tweak all the variables to make a very heavy thiol wine. But then you would end up using that as a blending component. It wouldn't actually be pleasant purely on its own, because this sort of easy to tip over into unpleasant sensory descriptors as well. However, the concentrations at which some of these thiols are active are insanely low. And some of the compounds of interest include sort of 4-MSP, 3-SH, 3-SHA, and 3-S4-MP. And why are they interesting? Well, the sensory thresholds for these are down in the nanograms per liter. So absolutely crazily low. And they have a lot of, at the right concentration, very pleasant sensory descriptors. We're talking about guava, passion fruit, grapefruit, even sort of blackcurranty, ribes, rhubarb, white grape, things like that. And so a lot of these can actually be free. They can obviously be found in free form in hops, and they can be found in bound form. One of the big challenges in this space for research is actually just the quantification of these compounds is incredibly hard. They're sensory active at such low levels that's a big part of the challenge of studying them, particularly in brewing, where you sort of have potentially more confounding factors than in wine. And to give you some idea of sort of how small these concentrations are, the default threshold for diacetyl is 100,000 nanograms per liter, and some of these are sensory active at 4. So yeah, orders of magnitude below it. So you can, as mentioned, you can add them in the free form, which is very popular, typically as a dry hop, and they can obviously also be modified by yeast activity. So 3-SH, for instance, which on its own kind of has a sort of grapefruity, sort of maybe lightly tropical character. When esterified by yeast activity and fermentation, it will change into 3-SHA, which, as you can see, sort of starts skewing towards passion fruit guava. Sensory threshold, again, you're looking at 10 times lower, basically. So, yeah. So bound thiols exist in hops as well, and in far greater quantity than free thiols. As mentioned previously, they're bound to amino acids in malt and hops. So typically bound to the amino acids cysteine, glutathione, and glycine, of which glutathione bound tends to make up the majority of bound. Okay, and so why does that matter? Well, glutathione bound are even harder to release than the cysteine bound. So we're only freeing a fraction. And I think the best way to think of it is the additional complexity in freeing thiols compared to terpene alcohols. That's because the release mechanism involves the non-volatile precursor having to be transported inside the cell. The precursors are then converted or liberated to volatile thiols using probably the beta lyase enzyme within the cell. And then those thiols need to be excreted back out of the cell into the wort or beer. So as you can see, there's a lot of steps at which conversion can be held up, or the final expression of it back into the beer can be held up. On top of that, these compounds are very, very volatile, so that it's easy to lose them with sort of more rough processing as well. So, and this is just sort of a rough idea for you, but this is a relatively thiol-heavy hop variety, and you can see that the free fraction of it is the blue, right down there at the very bottom, and then the cysteine bound would be the orange, and then the glutathione bound would be the green at the top. The colors aren't incredibly clear on the small screen, but so as you can see, if we can get yeast to free even a tiny fraction of either of those bound pools, we have potentially significant impacts on our final beer. That's why a lot of research is underway in this area. And as you can see, if you want to put numbers on it, conversion efficiencies are down at sort of 0.5%, or even lower, right down to less than 0.1%. So this leads on to another, hopefully very useful slide for brewers out there. It's something that our R&D team and research partners like Oregon State University have done some really good work into. There was a study published in 2023, which essentially showed some really interesting results. It showed us that not exactly as we had assumed, in that if a strain is better at freeing thiols, it's better at freeing all thiols. It's actually more so that certain strains are better at freeing specific thiols, which is not really something we expected. And this was a study which I'll show you the data from it in a second, but it allowed sort of the building of this, which allows far more specific recommendations for brewers, I think. And so, as you can see, we've done the same thing again, in that we have sort of done a big literature search and we've identified high bound thiol hop varieties. Again, this list is not exhaustive, which could be used sort of in your whirlpool or dry hop. Then we have specific strains which have shown greater ability to free that specific thiol, and then also hop varieties which are high in the free thiol. So then you can basically target your efforts to create these sort of sensory profiles that you want. Because it may be that you might want some of one of the terpene compounds, but you might want one of these styles as well. Sometimes it's crossover within varieties, or you can build your hop build to specifically achieve what you want. So quite interestingly, some strains that have been in the Lallemand Premium catalog for quite some time actually came out showing really quite high thiol freeing of specific types. In particular, Nottingham was a bit of a surprise, but it basically was good at freeing everything that we tested, which are obviously these. Interestingly, Diamond Lager was our top freer of 3SH from bound form, and also strains like Verdant and Voss showed some really good application for this as well. Again, very much like with citronellol production, if you want 3SHA in your final beer, you'll want to use hops that are high in free 3SH slightly earlier in the process. So potentially a cool whirlpool or a really early dry hop. And then we sort of found Nottingham was sort of tested very high for 3SHA. Actually, another one of our strains, Farmhouse, did as well, but it's POF, it's phenolic. So I wasn't sure about putting that one in just in case there's interactions there. And so this is taken directly from this paper. And so it's a really cool study. It was very real world, I would say. And essentially in this study, a wort was produced. It was split many ways, and fermentations were done with a number of Lallemand Premium strains. And so, I mean, you don't have to focus too much on this graph exactly, but what you can see is thiol precursors sitting on the right here, both in the worts and in the final beers, and then you can see the free thiols. And so this is by fermentation and analysis. And the analysis was done through a lab in France, Nicios, which is sort of has pretty good resolution on this stuff. And so for the brewers out there, the total hopping rate in this study was basically 2 grams per liter US Cascade. Sorry, I didn't convert that one for the Americans out there. US Cascade is a sort of early kettle edition, and then 3 gram per liter US Cascade pellets as a whirlpool edition, and no dry hopping. So the reason the Cascade was chosen is it's reported to have low free thiols and high thiol precursors. And so what you can kind of see the result is, you can see, I guess, some of what I've just highlighted. So Nottingham came up sort of really quite good across the books. Verdant would definitely say it was reasonably good across the whole range as well. And some really notable and interesting release by Voss particularly, and also Diamond. And also, as I say, Farmhouse, which is our non-diastatic Saison strain, showed some really great ability for esterification, which is about what we would expect. So on top of this, we can also consider process as well as ingredients, and it tends to be more important for thiols because they're a little bit more complex in terms of they already have very low release efficiencies. So some sort of broad brushstroke process considerations. Well, to produce more, these are some of the things that most likely will help to get a slightly higher conversion efficiency from bound. So one paper showed a really, really useful point for everyone, which is post-fermentation conditions are really important. As I went through earlier, you can see that the freed thiol is still inside the yeast cell and it actually needs to be released. And so the study found that essentially if you put it on chilling immediately after sort of fermentation is done, the yeast won't necessarily release those thiols. So they actually found that basically a 3 to 5 day maturation period at as low as 4 degrees then showed essentially the sort of peak release of free thiol back out of the yeast cell again. It's looking quite likely that warmer fermentations lead to higher thiol release. The only caveat I would add to that is that then you can potentially get sort of confounding sensory compounds being created as well, like higher esters, potentially higher fusyls, or even higher phenols in a phenolic strain. So I think the question still remains as to whether the overall outcome is going to be a lot higher tropical expression or whether it will also sort of be more complex than that. As yeast are creating beta lyase to affect these bound thiols, the main reason that yeast are interested is obviously for the amino acids. They don't care about us drinking delicious tropical beer. And so they will downregulate production of beta lyase if we have excessive fan levels in our worts as well. So your free amino nitrogens, which, it's hard to give really good recommendations in this, but that is one of the levers that could be pulled to potentially express slightly more thiol. Obviously not to the point of having bad fermentations though. And so I always like to think, being an ex-brewer, I like to think of retaining the compounds once you made them as well. So you can make as many thiols as you want, but if you have sort of downstream processing that's not going to be good at retaining them, well, you're going to lose them all again anyway. So some ideas might be to consider lightly pressurized fermentation or relatively early splitting at the end of fermentation to try to capture some of those volatile thiols in. I would definitely always urge a sort of very gentle post-fermentation handling. So consider your carbonation method, consider your transfer method if you're going to bright beer tank. These compounds are very volatile. They want to flash off, and they will if you let them. So, considering like top pressure carbonation and inline carbonation where they can't really escape from solution is a great idea. Obviously, as well, avoiding the use of fining agents, which are likely to reduce thiols, like copper finings. That's not copper as in kettle, that's copper as in copper sulfate. And I think this is probably a really interesting point in that, given that sort of lager yeast seems to have a pretty good ability for freeing thiols, whether it'll actually come out that way given the potential for sulfide production as well. And so just a little thought experiment before I sort of wrap up for the session, but I guess how would you then try to use this, all this resource to try to put together a recipe? Well, I'm from New Zealand, so you have to forgive me. I'm trying to target a sort of a cold IPA style with a New Zealand white wine type character. Sort of key in New Zealand white wine, some key thiols are 3S4MP and 4MSP. Very common in many aromatic whites here. So, all right, so we'll take those out of our handy flowcharts and decide on a very standard cold IPA grist of sort of 80% lager malt and 20% either rice corn adjunct. Then we can select hop varieties high in bound thiol. So I decided that I like the sound of Hallertau Blanc, Nelson Sauvin, and Simcoe for a Whirlpool edition. Then I'll select either Nottingham or Voss, which are good at freeing both of these thiol compounds. And then essentially decide on a fermentation regime, which will get potentially slightly more character. I mean, it's a cold IPA, so I still would like it to be quite clean. So I'd probably be fermenting Nottingham at sort of 15 degrees C or something like that. And then sort of assess, and you can top up with a late dry hop with any of these hop varieties as well. And I think that's kind of a very basic idea of how you would try to use this to direct your sensory outcomes in a certain direction. So yeah, I really hope you enjoyed the talk. If you're interested in accessing these sort of flowcharts that I've just gone over, they are available in the refreshed Biotransformation IPA Solutions best practice, which you can find a QR link to there. It's also linked in the Crowdcast as well, and that has a bunch of that information. It's got some other information as well. It's great. You should definitely get into that. And also there is a link on the right to our most recent, our previous Lallemand Brewing newsletter, which is a big biotransformation edition. Some great articles in there, and also some links for sort of recipe inspiration using our most recently released strain Pomona. And on that, I am going to wrap up, and I'm happy to take some questions. Thank you for listening.
Thank you, Scott. A lot of great information on practical tips and tricks for making better IPAs. Let's move on to take some questions. There's already some questions that have been asked, but again, if anybody else wants to add questions, there's a button, a question mark on the right of your screen. So you can ask questions there or upvote questions that you think you would like to have answered first. The main question a few people were asking is, will this be recorded? Yes, it is recorded, and we will post this on our YouTube page as well as through the original Crowdcast link. So no worries about that. Onto a more technical biotransformation related question. What would you do differently when seeking biotransformation in a low alcohol IPA compared to a typical higher alcohol IPA?
Because I'm guessing the implicit assumption here is that you would get less of it. Yeah, it depends how low you're
Basically
talking.
going with the trend for lower alcohol products. How in general, like, are there any different process or ingredient considerations that should be made for lower alcohol IPA styles?
Yeah, I think it definitely is going to depend on what you would categorize as lower alcohol in this case. I think if you're talking about like very sort of NAB lab range, like actually non-alcoholic or 1 to 2% ABV.
Yeah, let's go answer first based on kind of a session IPA, maybe
A
a
session IPA.
4%.
Okay. I mean, certainly in terms of thiol release, you probably won't see a crazily different or not a huge, like hugely lowered amount of thiol release. There was one study that's sort of compared thiol release across different work gravities. It wasn't hugely different between, I can't remember exactly, somewhere between 8 and 16 Plato. It sort of peaked a little bit in the middle, but it largely isn't that different. You're still getting a pretty significant amount of yeast activity in the production of like a session IPA. So, like realistically, you're going to get pretty similar release if you're looking for release of bound compounds in that range, even as compared to something stronger. And in fact, there's probably a good argument to be made that there's going to be less fan in those beers, in those worts. So you might actually get greater thiol release compared to something that's
Yeah,
sort of 8%.
I agree. Probably better, potentially more thiol release, but hop rates will tend to be lower in lower alcohol IPAs, so you'll have less aroma precursors to work with in the first place. But in general, not really any fundamental changes to the principles of
Yeah,
optimizing
correct.
biotransformation.
Correct. And I mean, you can consider yeah, you can consider it in a slightly different way, that perhaps you should focus on, in a beer like that, you should focus on using more of a higher precursor hop variety that has lower alpha acid. So even though you'd be targeting the same IBU throughout kettle usage to hit the same IBU, you'll use a larger physical volume of a lower alpha hop, which might, and then you can obviously select one with higher precursor to try to maximize that.
Okay. Do you want to speak to non-alcohol or maltose negative fermentations for biotransformation?
Yeah, that's probably a lot more challenging because
Yeah.
fermentation is extremely brief. You're only attenuating 10 to 20% of the wort sugars. Um, Obviously, that then impacts sort of the entirety of the yeast's metabolism. So it's fairly hard to do too much from that side of it. Um,
And again,
I think
hop
that's
rates are going to be
probably,
much lower in non-alcohol styles as well.
yeah, yeah, I tend to
I'd say
see,
relying on yeast metabolism in this case is probably going to be, it's just so limited because of the short fermentations.
yeah,
Maybe an
correct.
opportunity for using enzymes like aromazyme if you want to try to boost terpene levels a little bit. But probably your choice of just free aroma hop or alternative hop products that you add at the end of fermentation um, would probably have a greater effect.
Yeah, yeah. Getting sensory intensity and alcohol IPAs can be obviously challenging due to sort of the issue of just adding more, for instance, T90 hops and getting excessive polyphenol for the total palate weight.
Okay. I'm going to move on to the next question. Is there any benefit to mash hopping with high thiol precursor hops? I guess I might extend that question to include a discussion of precursors from malt as well.
Yeah, good question. I was guessing that someone was probably going to ask that question. I know that other yeast labs have published data on this, in particularly in combination with GMO strains. Basically, I tried to keep as much of my presentation to peer-reviewed literature as possible, and so there is no, not that I'm aware of, there is no peer-reviewed scientific literature that deals with mash hopping. I have no doubt it's coming, but unfortunately, I'm limiting myself to sort of what I can actually find in literature currently, which is nothing with mash hopping. Except, I mean, the only thing that could be relevant with mash hopping that there is literature on is the ability of mash hops to reduce oxidative potential of your IPAs. So there, it's going into the science a little bit, but mash hopping is thought to have some effect on chelating, binding up heavy metals such as iron and copper, which then aren't in the product during aging. And particularly when it comes to copper, it's going to preferentially sort of bind to sulfur compounds typically. So there's definitely some ability for mash hops to help even without GMO strains for oxidative sort of longevity, slightly better. That is definitely published.
Okay.
Uh, yeah. Sorry, was there a second half of that question?
I wondering if you wanted to speak to the presence of biotransformation precursors derived from the malt itself.
Yeah, well, that's a pretty cool topic area because it's definitely not something that was ever discussed previously. Was getting tropical characters out of malt, but there's been some preliminary work done which does show that there are, I think, particularly glutathione bound thiols in malt. Um, in particular, it would appear that the sort of most lowly kiln base malts tend to be higher in the precursors. So, for instance, in the cold IPA example I gave, I would look at using like the palest malt that you could for the 80% barley, which might help to sort of load up a few more precursors in your fermentation.
Okay. Moving on to the next question. A question about hop creep. Um, so um, not really covered yet in this presentation, but definitely related to yeast metabolism. How does the late addition of hops related to freeing terpenes affect the chances of getting hop creep? I say in general, can you speak a little bit
Yeah,
to um, how you would consider hop creep with regards to um, hop addition timing and process considerations and
I think I
biotransformation.
think I I think I understand maybe the edge of this question is that I was discussing bound terpenes are attached to a sugar moiety, so you're getting some freeing of sugars by that. Uh, that is overall as a portion of the weight a very very small fraction. So you wouldn't expect any additional fermentation from just the freeing of terpenes. Hop creep sort of is more to do with the enzymes which are still present in the hops. For instance, you can get alpha beta amylase and like amyloglucosidase activity from hop pellets or flowers. Um, yeah, and I would tend to see that as a separate process to, I guess, what I've just talked about. Does that answer the question?
I think so. Um, the next question. Let's see. When exactly would you add the early addition biotransformation hops in the fermenter? And I guess there's a few different options for early additions in order to optimize and increase the extraction of precursors and also give time for the yeast to interact with those hops.
Yeah. Yeah, I mean it's a great question. I think typically when us brewers sort of started looking towards biotransformation, it was all about early dry hops as a way to introduce these precursors. Um, that can raise other issues, sometimes regarding like sort of haze stability or also ferment vigor with the introduction of nitrogen from the dry hop, even early dry hop as well. I think a lot of breweries that are going for this now are sort of loading up high quantities as a like either a cool whirlpool or a just a regular whirlpool. Um, the bound precursors are I can't find a definitive reference for this, but I'm fairly sure that they're not that volatile. If you can't smell the
Yeah.
precursors, then they're probably not volatile. So you don't have to worry as much if you're just targeting bound precursors for even a regular temperature whirlpool. But typically, I would say a lot of brewers that are brewing these styles are targeting mostly whirlpool use now as well. I mean, if you're not worried about potential issues with sort of haze stability and stuff, you can add it in yeah, I mean, the first sort of 24 to 36 hours as you're rising towards peak cell growth, peak cell count, something like that.
Mhm. Okay. Moving on to a question about beta-glucosidase timing. So for exogenous beta-glucosidase enzymes such as aromazyme, are there any timing considerations for maximizing terpene alcohol release?
Yeah, it's a tricky question, actually. One of the things I should mention is that often enzymes are repressed by what they free. So, in the case of glucosidase, glucose will actually repress that enzyme activity. So it's usually not a problem because there's not much glucose in worts. But you'd probably be considering you could consider adding an early ferment. It really depends on when you're going to have those precursors available. So if you're sort of adding the precursors early on, like kind of what I've suggested, you could consider using it even sort of a day into ferment. Um, but if you're introducing a bunch of precursors sort of later in ferment with your dry hop, then you can add it with your dry hop. Sort of depends on where you're adding the precursors, really.
Okay. Next question, following up on the points you were making about cold maturation for increasing thiol release. So can you provide, in general, a little bit more information about the cold maturation at 4 degrees for further thiol release?
Just trying to remember off the top of my head. The study it was from a study from a year or two ago, and they were specifically one of the things that they were testing was whether any sort of maturation period was necessary for it. And so I think they found, and I'm not entirely sure why they chose 4 degrees. To my mind, post maturation temperatures that were fermentation temperature would do it as well. So I guess is some of that might be incorporated into sort of like a diacetyl resting period as well. But yeah, that I think based on that study, it also suggests that potentially, even given that little bit of extra time at that sort of low but not really low temperature, might help the yeast sort of just release some of that. From memory, it was sort of on a curve, like it kind of like 3 to 5 days was kind of optimal, but sort of past 5, it was starting to almost just, for some thiols, it would go back down a little bit, which I'm not sure, binding or something going on. But yeah, that's what I remember from the study.
Okay. I'm going to skip down to one question about dry hopping. Dry hopping at 5 to 8 grams. I'm not sure exactly what rate that is, but I guess in general, higher dry hopping rates can add a lot of harshness to the beer, and to which to mellow it down for 2 to 3 weeks, some of the aroma is lost. What can be done in this case to not have the harshness or hop bite, and at the same time, um, sorry, the question just moved on me. And at the same time, have preserve your hop aroma. I was wondering if you could take you could add your own comments, but maybe take a minute to introduce ISY Enhance as a potential for that.
Yeah, yeah, I think it's a great question that probably has existed from the dawn of hoppy styles, for sure. Same as when you brew hoppy lagers, right? Do you still lager it, or you just try to package it earlier? Um, I think it's always going to be on a sliding scale with the other components of the beer. Some beers, potentially higher ABV and things like that, are better able to balance higher polyphenol loads. Obviously, there are processed products as well in that space that can help by reducing the green matter. We released, was it last year before, a sort of yeast autolysate product called ISY Enhance. Initially, sort of potentially more focused at non-alcoholic beers. It's a sort of a specific fraction of the yeast cell that contributes body, and it can also sort of bind up some harshness as well. Eric, I think you've been a little bit more involved in the sort of release of that product. Do you have any sort of further comments on using that?
Yeah, it's a fraction of inactivated yeast that is rich in manoproteins that adds an element of body and mouthfeel to the beer, which is great for non-alcohol styles, as you mentioned. But for hoppy styles, it tends to round out some of the harsh flavors and hop burn you can get from higher hop rates. So allowing the aroma to shine through, but kind of taking the edge off of the harshness that you might see with higher hop rates. Um, in the UK in particular, right now, they're having great results with hoppy styles with this product. So it's something fairly new to the market, fairly new to the brewing industry in general, not specifically related to biotransformation, but certainly one of the tools that a brewer can use to optimize hop flavors.
But yeah, I guess having thought about it a little more, I'd say general sort of dry hopping best practice if you're using particularly T90s. A lot of the sort of polyphenol and harshness is thought to come from sort of the green material, the non-lupulin material. And I guess general best practices on that. I mean, there are people that are more knowledgeable that have written about it, but tend to limit in your dry hop period to sort of no more than 3 days, and making sure you're starting to get hops off relatively soon. From memory, the sort of studies that have been done on dry hopping, if you're actually agitating your dry hops and getting them into suspension, your extraction is largely done within 24 hours usually. That's sort of the thought. And, past that point, you're only increasing your sort of tannin and harsh characters. Again, that's assuming that you're getting good contact while the hops are actually in there. So that could be, CO2 rousing your hops, or some people pump them over, depending on what you're comfortable with, and then basically are trying to get them off as soon as possible after that. And I think
Okay.
one of the things to think about is, when you if you're a commercial brewer or a home brewer, you add a really high dosing rate of dry hops, and often it will, maybe just you're adding so much that it's all sort of sitting dry on top of the finished beer. So it's all about getting that initial integration into the work column or into the beer column, and then getting the extraction as fast as possible, and then getting rid of it. Basically, it's probably
All
the
right.
best.
I'm going to skip down a little bit to another question about the potential for biotransformation to reduce hop rates. Will biotransformation allow a brewer to use less hops in a batch and still reach the desired profile results?
It's something you have to trial and find out. There's definitely there's the potential to. I guess it depends how deep you want to go into the sensory profile of your products, really, or the beers that you like making. If you've got core recipes where you're trying to hit a particular profile, um, which might be, grapefruit or white grape or floral or whatever it might be. And potentially at the moment, you might sort of look at some of these charts and go, oh, well, actually, maybe not all the hops I'm using actually achieve that. So potentially there could be a refocusing, which would allow you to potentially do that. But yeah, no promises. You'd have to certainly experiment with it in your beers, and do some sensory to back it up in house to see that you can get that. But there's certainly the possibility of it will certainly change the profile of your beers for sure, depending on which way you target it. Yeah,
Yeah,
that's
I
probably the best
agree.
comment I've got.
I don't think you're going to be able to cut your hop rates in half, um, but
I
potentially you
Yeah,
might be able to reduce your hop rates a little bit, but I would put more of the focus, like you said, on modifying the flavor profile. One of the benefits of leveraging the tools of biotransformation, the way I see it, is that you can use a fewer varieties of hops and get different flavors by using the hops differently and combining them with different yeast and taking advantage of the power of biotransformation to change the flavor profile, not necessarily
yeah,
just to increase it. And in this way, by having fewer different bags of hops open, you make sure that the hops are always fresher. You don't have that one bag of hops that's half used and you don't touch again until you brew that beer again a few months later. Keep your hops fresher, use fewer varieties, and get a more diversity of flavors from those hops just by using them
yeah,
differently and leveraging biotransformation.
yeah. Good comments, Eric. And I think also, like, I guess to that point, hopefully the flowcharts kind of give you an idea of that you're actually chasing specific sensory compounds that at the moment you probably could be getting just in a free form from um, more expensive hop varieties. So there is always the possibility of then targeting your process around maybe less popular hop varieties that happen to be high in bound forms, and then sort of leveraging that biotransformation to try to get some of that character as well. So not letting one variety doing all the heavy lifting as a late dry hop.
Mm-hmm. Take a question about the type of hop product and how that affects biotransformation. Are T90 or cryo hops or any other type of hop best for biotransformation? Does the form affect the bound versus free terpenes and thiols?
That's unfortunately a question for a hop supplier. The all the literature that I read through was all based on T90s. So I guess that's the sort of synthesis of what I've presented. Yeah, is all T90s. Um, I'm sure there is some very interesting stuff to know in that space. Yeah, where obviously concentrated products are quite possibly higher in certain things, but unfortunately, yeah, I didn't
Yeah,
find any of that in the literature.
yeah, I don't really know myself how much of the precursors versus the free aromatic compounds are present in different types of hop extracts and whatnot. But
Mm.
precursors
Great
is
question
not
though.
the only. It's a great question. I'd be interested to know a lot more about that tomorrow. Or sorry, I say tomorrow because I have a meeting with a hop producer tomorrow to talk about biotransformation. So I'll ask the question. Um, but conversion of precursors into free aromatics is not the only type of biotransformation. There's also conversion of one terpene type into another terpene type. There's modification of aromatic compounds within a specific aroma type. Um, you would not have to worry about extraction to the same degree if you're using a hop extract because you're not having to extract the material from the hop material. So it might affect the timing. You can probably add it a little bit later because you don't have to worry about extracting it. I would say extracts, don't add them to the kettle, probably add them to the fermenter because you don't have to worry about that extraction. Um, but certainly an area that needs a bit more research. We still have a few more questions here, but I think we're already over the 1 hour point. I think that we have the ability to answer the questions after the fact. If you want to come back and visit the Crowdcast link, we'll make an effort to provide text responses to any remaining questions. So I guess the last question I'll answer is the presentation available? Yes, you could send me an email at brewing@lallemand.com, and I'd be happy to send you a copy of the slides. Those emails come to me, and if there's any additional technical questions or technical support, things like that, feel free to email me again, brewing@lallemand.com. Check out those QR code links for different biotransformation resources, or the learn more about biotransformation link at the bottom of the page here to have access to our biotransformation and IPA solutions best practices. And thank you all for attending the webinar. I hope you leave with a few more practical tips for making better IPAs. Thank you, everyone.